do you think of when you hear the phrase genetically modified organisms? Genetically modified organisms, also called GMOs, are living things that have altered genes from genetic engineering. Transgenic plants are one of the most common forms of GMOs. In this video, we will explore the history of and science behind transgenic corn. Expert opinions will be provided by the following professors at the University of Illinois at Urbana-Champaign. Dr. Fred Below, Professor of Crop Physiology. Dr. Stephen Moose, Professor of Plant Genomics. Dr. Rita Mumm, Director of the Illinois Plant Breeding Center. So why do we care about transgenic corn? Most corn grown in the United States is GM, or genetically modified. Because corn products are used in so many industries, including food, biofuel, and even manufacturing, you encounter a transgene every day.
The adoption by farmers of GM crops has just been very, very rapid over the last decade and a half.
In the U.S., almost all corn has some genetically modified trait in it. Corn is used in so many products that you probably confront in some way a transgene, every day in your life.
When did this all begin? The first major commercial introduction of GMO corn occurred with the production of Roundup Ready corn, corn that was genetically engineered to be resistant to the herbicide Roundup.
Roundup actually was not used in agriculture because it would kill all the crops and the weeds. So it was used around your garden and home, things where you would control where it was sprayed very easily. So it already had that market. But then someone had the idea when ag biotech came along, hey, we could put a gene in that could make it resistant. Where, like, for example, the Roundup Ready corn, that has a gene that allows it to resist Roundup. And that doesn't exist in any plant. So they got it out of a bacteria. There are actually some plant genes that can be modified to be resistant, but they require you take the gene, you make some changes in the DNA, and you put it back in. And both strategies have been used to do Roundup Ready.
So scientists were able to take a gene from a soil bacterium and insert it into corn to make the corn resistant to the herbicide Roundup. This meant that for the first time, farmers were able to use the herbicide Roundup to control weeds, and it would no longer kill the corn crops as well. The next major development in GMO corn came with the introduction of corn that was resistant to pests, such as the rootworm.
The first rootworm-resistant genetically modified hybrids were introduced in 2006. And before 2006, everything was insecticide or rotation. And I remember it immediately because all of a sudden we could routinely grow 200 bushels, where before 2006, 200 bushels was quite an accomplishment.
I was working in corn, and the American farmer was pulling to market GM crops, so very anxious to have in their hands insect-resistant corn, herbicide-tolerant corn.
The introduction of corn that was herbicide and pest-resistant led to much bigger yields for farmers. However, scientists are now turning their attention to more ambitious plans for GMO corn. The two biggest environmental factors that affect corn yield are the weather and nitrogen usage. Therefore, scientists are now working to make corn that is drought-resistant, efficient at using nitrogen, and yields a large, highly nutritious crop. However, these traits are multigenetic, meaning that they are controlled by multiple genes. This makes working with them challenging.
If you think about the sort of the next key biotech traits or the next key developments, they're going to be drought-tolerant corn, which is largely controlling the weather, and then they're going to be nitrogen use-efficient corn, which is going to be about getting as much yield out of the nitrogen you apply. And that's because those are the two factors that have the biggest impact on crop yield. Both of these characters, drought tolerance and nitrogen use efficiency, they're way harder than insect or herbicide resistance. Those are single gene traits. But drought tolerance and nitrogen use efficiency are quantitative or multi-gene traits.
True. It's many genes. They interact in ways that they actually interact differently when you have low nitrogen in the field or when they have high nitrogen in the field. So that makes it difficult. But then what do we do to overcome that problem? And I think one of the strategies, and the one that we're doing, I think it's going to be the effective, is you have to understand how those genes are regulated. When you understand the major regulators, the network, the complexity becomes much simpler. Like the more we learn, the more we realize how complex it is. And so we are better at it, and people do use information now. You can have some information about, like if you're going to do, if I want to modify a trait, I can look at, if I, like for example the protein, we have these examples of these very extreme proteins.
Oh, yeah.
We can go in and look at what other genes were impacted there, and we're doing that actually. So then if some of those are negatively correlated with yield, well then we'll know that, and we'd be able to, oh maybe we can get the protein but not the other things.
Yeah.
Because we know it, we can actually, oh I want this but I'm going to make sure this doesn't change. Yeah, like a circuit, we'll say. But if you knew where the major switches were, then that's where you'd be making the changes. But you would also have an idea of, if I do change it, what other things are going to happen? And I guess what's most important is, what is my range that I can do it and not have the negative impacts elsewhere, but make the change in the desired way. And so what we're trying to do in my lab is we're trying to understand the regulation of the nitrogen system, how the plant uses the nitrogen it gets. And part of that is these selection materials, because protein is the way the seed is storing nitrogen. The white and yellow seeds are just, it's a cross of two kinds, so that doesn't matter so much. But you can see these pink seeds on here, and that doesn't look natural, does it?
Not at all.
Okay, so what is being produced in these kernels is there's a gene we've put in. We didn't actually do it, another lab did it, as a way to study how the proteins in the seed are localized within the seed. So the seed, there's a group of proteins that are produced in the seed. They're the major ones. So when you say you get protein out of corn, there's a certain class of proteins that are the most abundant. The gene for that class of one of those kind of proteins has been, the promoter, the switch part of that gene has been fused to what's called the red fluorescent protein.
RFP?
Yeah, and it's from a coral.
Okay.
It's from a coral, and these coral, they live in, like, it's a little bit dark, so they create their own light by fluorescence.
Gotcha.
And so this red fluorescent protein has been fused to this switch that is only active in the seed, but very strong in the seed, because these are the most abundant proteins in the seed. And so that's what we have here. And so these kernels that are pink have that gene, and those that don't. And so what we can do is monitor. So now we know that this is a tool now to understand how this regulation occurs. We don't really know what the basis for it is, but we know it's different, and now we know that this gene is responding to whatever that is.
And then maybe you go in with some of those more biotech computational tools and try
Yes, so what we're trying to do now is we're actually trying to map the genes that influence the pinkness.
Excellent.
And so we grow a group of corn. They vary in their pinkness, like you see here. And then we actually take a picture of them with the high resolution camera. It can extract out the red pixels and put a number on it, and it's more sensitive than our eye. And so you get a number, and then you can do that, and you can then relate what genes are associated with high or low pinkness.
Because RFP is bound to the major proteins of the corn seed, one can clearly see what seeds have the most protein by looking at which seeds are the most red. By examining the corn seeds on different plants at different points in their development, scientists can see what proteins are expressed, when they are expressed, and how strongly they are expressed. This allows scientists to study the regulation of the RFP-tagged protein and begin creating gene circuits. It's a simple way to study a complex multi-genetic trait, like nitrogen efficiency in this case. This shows a clear intersection of crop biotechnology and synthetic biology. As the two fields begin to overlap more and more, it will be possible to create new types of genetically modified corn for more complex traits.